Okay, and welcome to the monthly webinar hosted by the International Society of Biopharmaceutical Statistics, ISBS. We are very thrilled to have Professor Ying Yuan with us today. Professor Ying Yuan is a Bayesian Mori Distinguished Professor and Chair of the Department of Biostatistics at the University of Texas MD Anderson Cancer Center. Many of us, I think you know, Dr. Yuan is an international renowned researcher in innovative Bayesian adaptive designs, having developed numerous innovative designs for early phases trials, seamless trials, biomarker-guided trials, basket trials, master protocol trials. Today's seminar is repeated Bayesian interim analysis consequences free. I think it's especially timely in light of the FDA's this January 2026 draft guidance on the use of the Bayesian methodology in clinical trials. I think it will help clarify several open questions from that document. I just want to know during the presentation, please feel free to leave your questions in the chat, and we will address them during the Q&A session at the end of the presentation. And we will also share the slides and the recording with riches to the participants after the seminar. Without further ado, I would like to invite Professor Yuan to start his presentation. Thank you.
All right. Yeah. Good morning, and thank Haitao for the nice introduction. And ISBS organized this seminar. And we selected the seminar date 6 months ago based on availability. The timing, has turned out to be perfect. And as Haitao mentioned, shortly just after FDA released Bayesian guidance. So many of us, I think, may already read this guidance. This is a really, truly game changer and significantly expanding our toolkit, enable more efficient trial development. And if you will ask me which part of this guidance it's most exciting and paradigm shifting, and I will point to section 4. Because regardless a specific application, so like in section 3, describe a lot of cases can be applied Bayesian method, or prior you used as described in section 5. The ultimate goal it's always the same, right? So to make scientific and regulatory decision based on the observed data. The scientist must to make decision if the treatment is effective or not, and the regulator must decide approval or not. So this naturally leads to a fundamental question: what's success criterion? And does that success criterion support a robust operating characteristic and decision making under all possible scenarios, which is the operating characteristics? So this is a paradigm shifting because the guidance formally endorsed use Bayesian success criterion for regulatory decision making. And one of common used decision criterion and also described in guidance as example is this posterior probability-based criterion. So here, theta 0 is the minimum beneficial treatment effect. And you can think it's, for example, response rate of a control arm. And the theta is the treatment effect for the drug under investigation. And given the data, we calculate what's the probability the treatment of the new drug is better than a certain benchmark, like a control response rate. And if this probability greater than a certain cutoff, and then we take conclusion the trial is success, and the treatment is effective. And you can see in this criterion, the key parameter is this cutoff. And the guidance also outlined different approaches to calibrate Bayesian success criterion. And in this specific example is the cutoff C. And we can choose C to calibrate to a certain level of type 1 error rate. So this is more like a classical frequentist-based calibration. And the guidance also provides flexibility. We can use Bayesian approach to determine this cutoff. And for example, we can directly leverage the interpretation of this cutoff to select its value. Or we can also use a benefit risk assessment or decision theoretical approach to determine the C, or in general, success criterion. Another very important key takeaway from guidance is it underscores the importance and the necessity of simulation-based operating characteristics assessment. And obviously, one of the most significant development in this guidance is that it allows flexibility of use either frequentist or Bayesian operating characteristics to validate your study design, to demonstrate your study design can achieve a scientific conclusion. And also the guidance provide a very clear description on specific metrics tailored for each framework. So for example, if we are interested in a frequentist operating characteristic, and we can show the design have well-controlled type 1 error and the power, and also study the bias, mean square error, and coverage rate. So this approach is ideal for Bayesian method calibrated to type 1 error rate. So, but this is not the only approach, and we can also use Bayesian OC to demonstrate the sufficiency and robustness of the Bayesian design. And we can consider probability of correct selection and Bayesian power, bias, mean square error, and the coverage probability. So you may notice here, especially for some inferential metric, seems the same between the frequentist and the Bayesian. But there is a fundamental difference is when you calculate bias, mean square, and the coverage probability, in all those simulation-based assessment, the theta is random. So that means when you generate those operating characteristics, you simulate 1,000 trial. In the frequentist approach, the theta is fixed. For example, you set the non-value or alternative value. But in the Bayesian approach, in this 1,000 simulated trial, every time we draw theta from its posterior distribution. That means every simulated trial gonna have a different value of theta. So that's why if you look carefully at the guidance, when they describe bias, mean square error, they use the average bias, average mean square, and so on. But in here, we just call it a bias, mean square error, ignore the average. But keep in mind, so this is the Bayesian OC. That's why we have to consider theta as random. And this approach is suitable for Bayesian methods not calibrated to type 1 error rate. Okay, so today we will tackle one of the most intriguing questions in Bayesian adaptive design. So that is under the Bayesian success criterion, how do interim analysis with superiority stopping influence the OC? And this question it's generally understood in a frequentist framework. Sometimes we call it multiplicity. And we know when we take a multiple look at the data, and we're going to inflate type 1 error. And many well-known methods have been developed to address this issue, like operant framing methods, alpha spending methods to control the family-wise type 1 error. However, for Bayesian, there is no clear consensus. And some argue Bayesian methods adhere to likelihood principle. So therefore, they are not subject to multiplicity concern. So in other words, we can use posterior probability to make a decision as frequently as desired without any adjustment. So if we back to the posterior decision rule we described previously. That means we can use the exactly same value of C to make a decision, no matter you look at the data once or you look at the data many, many times. And at the same time, some researchers caution that Bayesian multiple looks can inflate family-wise error rate and also distort the coverage probability. However, those those counter arguments in the literature focus primarily on frequentist quantities, especially family-wise error rate. So this raised concern about their relevance and appropriateness of using frequentist metric to assess Bayesian inference. So that's created a lot of debate regarding this issue. And also in FDA also acknowledged a similar issue in its adaptive design guidance. And it stated relative to a family-wide error rate inflation, bias estimation in adaptive design, it's current a less well-studied phenomenon. So in other words, we should not only focus on family-wide error rate, we should also look at the impact of multiple look for the inference. So for example, the bias, the coverage, the mean square error. So that's exactly a goal of this presentation and this study. So we want to provide a theoretical justification and the numerical evidence on how Bayesian interim analysis influence the OC. And we also want to reconcile the conflicting arguments regarding the Bayesian multiplicity that have long persisted in the literature. So specifically, we want to clarify when and what statistical property that are invariant or varied to sequential look. And this is a joint work with Sui Liu at MD Anderson, Beibei Guo at LSU, and Laura Thompson and Lanier at FDA. Okay, this is a setup, and we consider a single arm superiority trial for simplicity, but the results are applicable to randomized controlled trial. And we use X denote the efficacy endpoint for the ICE patient receiving the experimental treatment. And we assume XI follow a certain distribution denoted by F, and the theta represent the treatment effect of interest, and possibly along with other nuisance parameters. And we, for simplicity, we assume a large value of theta indicate a higher efficacy. Okay, since we consider a potential interim analysis, and we use a small n indicate interim sample size, and the dn denote the interim data. And we use this posterior probability-based success criterion as also described in a guidance. And if given the interim data dn, if the posterior probability theta greater than theta 0 exceed a certain cutoff, and we claim success. And in a multiple interim analysis setting, we're going to stop the trial, claim superiority or success. And in order to isolate the effect of multiple look, and in here, when we compare so-called a fixed design, which means the design without interim look, okay, we just do the trial, finish it, and then we apply this Bayesian success criterion to make a decision if treatment is effective or not, versus the adaptive design. So during the trial, we take a multiple look, and but in those two comparisons, and we use exactly same value of C. So in other words, when you see any difference in the operating characteristic, that can be contributed to the interim analysis, rather than you use a different cutoff, which also will impact operating characteristic. So therefore, our results are invariant to the methods of determining C. So later on, we will describe a specific way to calibrate and select the value of C. But as I highlighted here, it doesn't matter what methods you use. And you want to calibrate type 1 error, determine C, that's okay. You want to use Bayesian methods like direct interpretation or any other method determine C, that's totally okay. And our results are invariant to that because we use fixed C, no matter you have interim or no interim. What we are concerned here is the difference between those two designs in operating characteristics. So again, as I mentioned, so our selection it aligns with what described in guidance, especially the criterion we used is based on this Bayesian posterior probability based criterion. Okay, and in here we focus on Bayesian OC and aligning with FDA guidance. And the other reason we choose this is to bypass the debate regarding the relevance of frequentist metric to Bayesian design. And so actually in general, our finding also apply to the setting if we use frequentist OC to calibrate the design. But again, in here, we focus on Bayesian OC and to avoid unnecessary debating. Once we use a Bayesian framework, and then we have to distinguish two types of prior. And one is data generating prior and the user specified prior. And that's what we call in our paper. And in the guidance, they are called design prior and analysis prior. So what's data generating prior? When you evaluate operating characteristics, we do simulation, we generate data. And in this setting, in order to generate data X, we basically do two steps. The first step is we generate a CEDA random draw from its prior distribution. So this represents the true distribution of the treatment effect across the population of trial. And once we draw the CEDA and we condition on it, we draw the data, right? So this complete one simulation. And if we do 1,000 simulation, we're going to draw 1,000 CEDA from this data generating or design prior. And conditional on each CEDA, we generate data. Of course, in practice, we don't know what's the true mechanism generated the treatment effect across the trial population. And when we observed a specific trial data, what we do is we specify a user prior, right? FDA guidance called analysis prior, and we do Bayesian analysis. So in this specific case setting, we calculate posterior probability theta greater than theta 0, and to decide if it is a positive study or not. Should we stop the trial during the interim or not? And so the prior, so this user specific prior going to use it to get a posterior distribution. And as demonstrated in the bottom, so it's just very simple base rule to calculate the posterior distribution. And to study the impact of interim analysis, and here we explicitly distinguish between the scenario where the data generating prior match the user specified prior, or you can say the user specified prior is correctly specified, or the more common case, they are differ. And that's also, this issue also discussed in FDA guidance. So if you look at the section, trials not calibrated in type 1 error rate in the operating characteristic section, and the guidance specifically mentioned, the user should study and demonstrate the probability of making incorrect decision is very low, even when the design and analysis prior don't match. Okay, so once we set this up, and the next is we need a performance metric. And in here, we distinguish two classes of metric. One is a statistical inference related metric. And in here, we studied the bias of posterior mean. And in other words, we use posterior mean as a point estimate of the CEDA. And we want to see how it different from the true value. So in here, we give a specific definition. So you will notice here, I highlighted the Bayesian framework consideration here. So when we take expectation, we not only take expectation regarding the data, right? That's the frequentist way to calculate the bias. But in here, we also consider the randomness of CEDA. So in other words, the expectation, it's taken over both CEDA and data. Okay, so that's one thing I want to highlight. And also similarly, we can define a mean square error again, so this is a Bayesian framework. We have to consider both randomness of theta and the data. And we also look at the 95 posterior credibility interval coverage. And those performance metric and also highlighted in FDA guidance. And in here, I highlight here, the guidance uses the term, I will say it's very precise term called expected bias and the expected mean square error and the expected coverage probability to highlight and distinguish it from a frequentist bias mean square error and coverage. So in this setting, the expectation is used to highlight the randomness of theta, not only data. Okay, so that's a set of metric which measure the statistical inference. And for regulatory and also scientific decision, another very important metric we should consider is a decision error. Because at the end of the day, the regulator needs to make a decision, approve or not, right? And the researcher typically have to make a claim the study is positive or not. So it's going to be a binary decision. So it's important, very important to measure such a decision error rate. So in here, we first look at the first discovery rate. And in here, we look at the two slightly different first discovery rate. And the first one is the standard definition from the original FDR definition. And the second one is called positive FDR, PFDR. You will notice the difference is very minor. And it's only depends if you have this last term, probability R greater than 0. So here R is the number of trials that claim the treatment effective. So that means the positive study. Or you can, from a regulatory perspective, it's the drug. Regulator believe it's positive, gonna approve that drug. Okay. And the V denotes the number of false claim where the treatment in fact not effective. So that is the error, right? So that is called the false discovery. And in here, we won't spend much time to distinguish those difference between those two FDR. And in general, PFDR have much better Bayesian interpretation. And the reason original FDR have second term probability probability R greater than 0 is to address the issue. Remember, original FDR is still p-value focused. And its goal is to use a step-up p-value procedure to control the FDR. And if you don't have the second term, and when all the hypothesis is none, and the false discovery rate always to 1. So that means you cannot, through a step-up p-value based procedure, to control FDR at a specific level. So that's why in the original FDR paper, they consider this definition. But actually, when you think about it, most people understand FDR. They more kind of thinking in a PFDR way, right? So among the positive study, how many trial or treatment effect, it's truly effective. So that's the some historical context for FDR and PFDR. And in the guidance, it does not specifically mention about FDR, but it does talk about the importance of probability of correct decision. So if you look at the definition of probability of correct decision, actually it's the 1 minus PFDR. So this is the definition of a probability of a correct decision in guidance. It's calculated as the proportion of trials where a positive treatment effect was present from those trials that concluded effectiveness. So you can see it's exactly 1 minus PFDR. But again, the difference between PFDR, FDR, it's pretty minor, okay? It's just, you can see it's different by a certain constant given a specific setting. And also guidance talk about a concept of false positive conclusion. And it's essentially it's the FDR or FDR, and it's also highlight this is a metric more suitable for Bayesian framework because it's conditional on a positive conclusion which is made on the data. So that means we condition on data rather than condition on the theta. And when you look at the a type 1 error, it's opposite, right? We condition on the none and how likely you reject the null hypothesis. Okay, so FDR and it's of a substantial practical interest because in some sense it can be interpreted as a risk of regulatory regret. And if we assume the positive results leads to a drug approval. So when you look at this definition, and in the denominator, it's number of positive study, right? If we assume each positive study reached the Bayesian success criterion, going to lead to the drug approval, and this is going to number of a drug approved based on the Bayesian success criterion, and the V is number of ineffective drug, right? So in other words, this quantify among all the approved drug, how many drug are not effective. So we wanted to control this regret to a very low value. So although FDR and another related point is FDR was originally developed as a tool to control frequentist error rate. Story showed that especially the PFDR, it's have directly a Bayesian interpretation. So in other words, so it's a very suitable metric to use to measure decision error for the Bayesian design with Bayesian operating characteristics. Okay, in here we also look at another metric called average type 1 error, and which is defined as the probability of claim effectiveness when the treatment actually is not effective. So in our setting, in here, the effect not effective means the treatment effect is less or equal than theta 0, right? And we look at how many, given this is true, and how likely we conclude the treatment is effective. And you take expectation. So that's the average type 1 error. So again, why we call it average? Because in here, the expectation gonna average across the theta. So in contrast, in the frequentist approach, and you're going to conditional on the null hypothesis. That means the theta equals theta 0. It's a point mass. Okay, you condition on that, and then you do a loss simulation. But in here, the theta is a random. So we're going to simulate the theta under the constraint. The theta is less or equal than theta 0. So that means when you do 1,000 simulation, every time you get different theta. And then you determine if you make a positive conclusion or not. And in frequentist, you always fix theta, for example, the 0. And then you do 1,000 simulation, only simulate data, and to calculate the error rate. So this average type 1 error, it's a Bayesian counterpart to the frequentist type 1 error rate. And sometimes it's also referred to a Bayesian type 1 error rate. And actually, this is not totally, new. It was described in FDA guidance for the use of Bayesian statistics in medical device clinical trials as an alternative definition of a type 1 error for Bayesian methods. So in the guidance, in the new guidance, it does not explicitly mention this concept, but it describes a concept closely related. So in here, the guidance talk about the Bayesian power, right? So it's a probability of meeting success criterion average over a prior distribution. So at the same time, the guidance also talk about the design. When we study this Bayesian power, we should also consider the prior, pessimistic prior, which means that prior essentially assume the treatment is not effective. So when you use such a pessimistic prior, essentially Bayesian power gonna becomes the Bayesian type 1 error or average type 1 error, right? And on the other hand, if you consider optimistic prior, Remember, this Bayesian power is average or prior distribution. So it depends on how you specify this prior. Again, this is a design prior, okay? So it's the true distribution of theta. If you use the optimistic prior, so that means the prior it's focus on the area, the treatment is effective. So in the example, it's gonna theta greater than theta 0. And this Bayesian power gonna very similar to the frequentist power, which means given the treatment is effective, how likely you're gonna have a positive study. So in some sense, actually, this is one of the comments, about this guidance. Personally, I think that point should be made a little bit more clear. And also the power, use the term power actually is a little bit confusing, right? So if your prior distribution cover both the support, the treatment effective and not effective. So this power essentially just means how likely you conclude the trial as positive. But it's the interpretation is not that useful to evaluate the operating characteristic because you don't know if that treatment is effective or not because the theta is random. So it's just how likely you get a positive study. And potentially it's confusing in the sense most people think about the power. It's thinking when the treatment is effect, how likely I'm going to detect and that effect in a positive study. Okay, so I will first present our findings and in terms of the impact of interim analysis, and later on I will show you some theoretical insight why that happened, and also some numerical example to show this phenomenon. Okay, so here is the results, and when we have matched prior, so that means the user prior is exactly same as the design prior, or analysis prior exactly same as the design prior. And when we have multiple look without any adjustment, that means we use the same cutoff, same success criterion as the design, fixed design. We don't have any look. And in this case, we always have unbiased estimate. And also we have nominal coverage. And so this in Bayesian terms, so those are consistent with likelihood principle, which means the only observed data matter for inference, not the stopping rule. And so those properties support the consequence-free claim for those specific metric. So this is a very amazing results, right? Because most people thinking is like this: when you have an interim look, when you see positive results, you stop. That seems to create a bias, right? Because when you see good results, you're going to stop. When you see bad results, you're going to continue until you reach the positive results. And similarly, because that most people will think the coverage is going to be destroyed. It's either over or under coverage. But in this case, okay, when the design prior exactly match the analysis prior, it doesn't matter. You can take as many look as you want. When you get a positive result, you can stop. You always get unbiased estimate and the nominal coverage. And however, when the analysis prior differs from the design prior, I will argue that's the most common case, right? So when you specify prior, you never know what's the true distribution of the theta. So in this case, our intuition is correct, okay? So it's going to create the bias for posterior mean estimate, and the coverage cannot guarantee a nominal value. It's either under or over coverage. And those properties supports the caution in literature that Bayesian multiple look can distort estimation coverage probability. Okay, so in here, my goal is not to go through mathematics, but because this results, it's not that totally intuitive, as I mentioned. Because intuitive, we think when you see positive results, you can stop. That will bias your sample, and eventually you will get a biased estimate. Why in the case when your analysis prior match the design prior, it doesn't matter. It won't have any impact. So here I just show this some mathematics so you can have more insight why that happened. Okay, so this is the definition of the bias. And in the second equation, essentially, we just plug in the definition of theta hat. Okay, that's the posterior mean. As I highlighted here, in here we use a Bayesian framework. So that's why outside expectation, we take expectation over both the prior distribution, the randomness or distribution of theta, and also the data. Okay, the pi g, again, reminder, the pi g denoted the data generating prior, and the pi is the user specified prior, which corresponding to the design prior and the analysis prior in FDA guidance. Okay, right now, when we, under the condition, the analysis prior is identical to the design prior. And when we look at this expectation, and we have several expectation, right? So when they are the same, we just can replace this user specified prior by the design prior. And right now we have exactly same distribution in this internal and external expectation. And by the law of total expectation, and it easily can seen, this will be go back to the true posterior mean of theta. So that's why eventually you get unbiased estimate, even you have interim look and stop. But when the analysis prior differs from a data generating prior, and we cannot, this law of total expectation does not work. So we will get a biased estimate. And similarly for coverage probability, and in here the I indicated if the theta is in the Bayesian credible interval, and essentially we want to take a look on average if this coverage can maintain the nominal value. And again, you have to take expectation in terms of both theta and the data. Okay, and when in the case the analysis prior match the design prior, data generating prior, again, you can do a very simple calculation, and you just replace this prior you used to construct the confidence interval or credible interval. And because they are matched, you can replace by pi g. And this exactly becomes 1 minus alpha. And by definition, because when you construct the credible interval, that's how you construct the posterior credible interval. And so in this case, you can exactly maintain nominal value 1 minus alpha. But however, when those two prior are different, and you cannot do this operation. So in this case, that's why it will deviate from the nominal value. Okay, so for those you are kind of more familiar and comfortable with this kind of mathematical reasoning, and you can see the difference when you have matched prior and unmatched prior. But some people still have struggling, right? And intuitively, when you stop, you have biased data. How likely, even the prior match, why it's still unbiased? Why the coverage is going to be right? So intuitive way explanation is this, okay. Again, we are in the Bayesian framework. The theta is random. So every time you take a different theta, right? And the theta can be very big. That means very positive study, very big treatment effect. Or it can be very small. That means the negative study. In here, I mean the truth, okay? The drug is not effective. So right now, when the theta is small, that means the drug actually is not effective. And when you take an interim look, whenever you see positive results, you're going to stop. Yes, for those simulated trial, it will overestimate the treatment effect. However, because it's random, right? So across 1,000 simulations, sometimes you're going to have a random draw of theta, which corresponds to a very big treatment effect. So remember, in this case, if you see the positive results, your Bayesian success criterion satisfied, that posterior probability greater than C, you're going to stop. So in that case, you're going to underestimate the treatment effect. Because if you continue the study, the true theta is very big. and the data eventually going to support a very big theta. So that means those two parts kind of canceled out when your data generating prior exactly match your analysis prior. That's perfectly canceled out. So that's an intuitive way to understand why, in this case, it seems a magic, right? You look at the data, it doesn't matter. That's how easy way to understand it. Okay, for mean square error, it's less interesting because interim look always increase mean square error, no matter the prior is correct specified or not correct specified. So in the case the prior correct specified in the sense the analysis prior exactly match the design prior, we know it's unbiased, right? So this one is 0. But the question is, because of the early stop, your sample size is going to become smaller. That means when you compare with the fixed design, you always go to the end, apply the Bayesian success criterion. You will have a large variance. But it's not an issue, right? It's not caused by any bias or incorrect behavior. It's just the early stopping leads to small sample size. But when the prior are unmatched, and in this case, the first term going to have a bias, and also second term, because the potential early stop, you have a large variance. That's why overall the mean square error gonna becomes larger. Okay, how about the decision error metric? And in here, it's different from inference metric. So in here, we even with a matched prior. So that means your inference or your analysis prior exactly match the data generating or design prior. The FDR, PFDR, and average type error are always inflated by interim look. So this is remarkable, right? So originally when we have matched prior, seems everything should play out well. There's no bias, no matter how many times you look at it. And there is the coverage rate have perfect coverage. But in this case, when you look at the decision error, it's different story. Okay, it's always inflated by interim look. And in here, I give some intuitive reason because mathematically to show that it's quite complicated. And if you're interested, you can look at our paper. And the reason for it's a little bit easy to understand why the average type 1 error inflated. And again, it's because the multiple look increase the probability of stopping on random noise. And but for the FDR, it's a little bit subtle because you have remember by the definition, you have two parts, numerator, denominator. And when you have early stopping, when you have positive data, and both are the number of discovery and the number of false discovery, both are increased. And because they increase at different rate, that's why overall this FDR will be inflated. But again, here I just give you a quick intuition. And if you are more interested in rigorous reasoning, you can look at our paper. Okay, so this results explain why conflict and argument exist in literature. So both sides are partially right. So the Bayesian monitoring is consequence-free for bias and coverage only if the prior is correctly specified, and it never consequence-free for decision error rate. And from a practical and regulatory perspective, the implication is that, and in practice, I think other than the case, we use informative prior to borrow information. And most of the time when we use Bayesian methods, we routinely use a vague or non-informed prior. And typically it's going to make this issue worse because most likely when you use non-informed prior, it won't match the data generating or design prior. And in other words, in this case, we most likely subject to both bias and distorted coverage for a credible interval. And also we know the first discovery rate and always inflated, and also average type 1 error. So that means in this case, multiple adjustments are necessary to safeguard the regulatory regret rate. Okay, so in here, I just quickly show you some numerical results to demonstrate the conclusion we just described. And in here, we consider a trial with a sample size 100 and with 2 interim look at 40 and 70 patients. And we use this Bayesian posterior probability criterion to claim success. And if this criterion is satisfied, we're going to stop at the interim. And in here, we calibrate the C such that under the case, the analysis prior is correct specified. We control the positive first discovery rate at 5%. And again, as I mentioned, it doesn't matter how you calibrate C, because in here, we are not focused on the absolute OC of a specific approach. We want to compare the design with and without an interim look to evaluate the impact of interim analysis and the superiority stopping. And again, when we simulated the trial, we follow the Bayesian paradigm. We first simulated seed from design prior, and then we generate data conditional on the seed. And we compared the adaptive design with the interim look and without interim look. Okay, so in here, I just show the results for the binary endpoint. We also consider continuous and time to event results are the same. So in here, we assume the true response rate of experiment arm generated from this beta 3-3 distribution. So again, this you can think this is a design parameter or data generating prior. And we set the response rate for the control, historical control, as a 60%. So in other words, we evaluated the posterior probability theta greater than 0.6. And we consider both matched and unmatched analysis prior, and we simulated 50,000 trials. Okay, so here is a quick look at the results. So here is the analysis prior. Okay, when the analysis prior is beta 3-3, that exactly match the design prior or data generating prior. And in this case, we look at the 2 designs. The one is no interim, the other is we have 2 interim look. And when you look at the bias and the coverage, so this is exactly as the theory predicted. You don't, even you have interim look, it doesn't matter. Okay, you have no bias, and also you maintained coverage at the nominal value. So when you look at the other case, this is kind of more common case. Let's say we want to use a non-informed prior, beta 0.05 and 0.05, which have the prior effective sample size 0.1. And in this case, you will notice the difference between the design. You have no interim versus you have interim. And right now you can see the bias, right? And also the coverage rate deviated from the nominal value, 95%. And for the discovery, first is FDR and average type 1 error. And in this case, you can see no matter the prior, it's correct specified or not correct specified. When you compare the design with interim versus no interim, the first discovery rate always inflated. It's almost double. And similarly for the average type 1 error rate. Okay, so this is a final slides. In conclusion, the consequence-free claim, it's valid in estimation and coverage only when the user specified prior perfectly match the data generating prior. A condition, unfortunately, it's seldom met in practice. And more interestingly, this consequence-free claim failed for FDR, or in the guidance is called a probability of correct decision and average type 1 error. Sometimes I know some people hate type 1 error. You can say it's a Bayesian power, use pessimistic prior. But anyway, point is not to argue it's the best metric or not. In here, we just show how it impacts on those metrics if you are interested in those metrics. So the conclusion is Bayesian methods are powerful and highly flexible, but they do require careful design and calibration, which I think is one of the point repeatedly highlighted in the FDA guidance. So in this specification, if we want to use Bayesian interim look to make superiority stopping, and the first is we have to adjust the decision threshold to account for multiple looks. And also we need to perform through sensitivity analysis to evaluate the impact of prior misspecification. So in here, misspecification in the sense how much your analysis prior deviate from the design prior or data generating prior. That's also highlighted in FDA guidance. So lastly, it's important to do a comprehensive simulation to evaluate the operating characteristic, no matter it's Bayesian or frequentist. Okay, so if you are interested in more detail, and this is the reference, and I think that's all I have today, and thank you for your attention.
Okay, thank you very much, Professor Yuan. I think it's very, very important talk and also very important research, clarify many, maybe confusions, many unclearness some people just saw from the recent FDA guideline. And I saw there is a lot of questions, but I think maybe we cannot have all be answered. But if I can all read of all your questions, maybe just allow me to ask questions, maybe. Yeah, I think it's also, I totally agree that I found it particularly interesting in the new guideline is the FDA explicitly opened a second pathway or category, and where Bayesian methods may be used for regulatory decision making without fully calibrated frequentist type 1 error control, but mentioned based on the totality evidence. So basically, just like you said, FDA not only just interested in the single trial posterior question, but also pay greater attention to cross-trial reliability. That is the reliability of the set of the trials that are declared successful when the same Bayesian decision rule is applied repeatedly across many trials. So this is just the Bayesian positive, false positive, and also the FDR that you mentioned to focus on today. I think it's very, very important. So I think this is maybe one of the first time this distinction has been explicitly placed on the table in the regulatory context. So could you comment on how this distinction should be defined, evaluated, and quantified in practice? I think some questions are related to this, and what implications it has for how we use Bayesian methods going forward in clinical trials and drug development. Even the first pathway exists there, we usually used already. And maybe a very another question is, I just wonder is that in the second pathway, the FDA didn't mention explicitly the average type 1 error. Although you pointed implicitly mentioned in this guideline, average the type 1 error is mentioned there. But I just wondering why the average type 1 was described in the 2010, the Bayesian guidance in medical device, but not explicitly in this guide. Maybe this is just a wonder, but yeah, this is my question.
Yeah, so I can answer your later question first. Of course, I don't know, how FDA is thinking when they drafted this guidance. That's my personal interpretation. I think a lot of time, again, a lot of Bayesian totally against the so-called type 1 error, no matter it's a Bayesian type 1 error or frequent type 1 error. Whenever they see kind of allergy to that term, type 1 error, I feel that way. But again, my personal view is if you consider Bayesian type 1 error, again, I think a lot of people against it is because by definition, it's conditional theta, not conditional data. But in the real trial, what you are interested in is given data. how data tell you about the theta, not given the theta, how likely you make error decision. So I think that's philosophically that's a lot of people against it. So my view is yes, it's not perfect metric, but I feel it's reflect a certain aspect of your trial operating characteristic, and if that metric totally wrong, totally out of boundary, definitely it's a red flag. But do you need to strictly control that average time error 5% or 2.5%? That's the issue can discuss. But I do think it does provide some useful understanding the methodology, the Bayesian methods, and it can be useful a safeguard, right? If that value is too high, definitely it's a red flag. But I won't argue that will be equivalent to a frequent time error every one. We have to control 5%. That's my interpretation of that. And I see, in the chat, there's a question say, the presentation or the paper mix two ideas, which create a greater confusion, choice of prior, and the sequential assessment. So I want to clarify that's exactly when we evaluate the impact of sequential assessment. We divided two scenarios. Why is the prior matched? Why is not prior matched? So in some sense, you can think it's a 2x2 table evaluation, right? So you can think one dimension is prior specified, correct specified, not. The other is you have a sequential assessment, no sequential assessment. And our conclusion is exactly as layout in that way. When the prior are matched and the sequential assessment have no impact on some inferential metric like bias, mean square error, but it does impact the first discovery rate. And when the prior does not match, in this case, it impacts both inferential metric as well as error rate metrics.
Yeah, that's great. And yeah, there is a lot of questions. I think maybe we can just pick one for the final. I'm sorry for the
Do
other
you mean the
questions.
chat? In chat, I cannot see much.
Yeah, a lot of kind
Oh
of
yeah, a lot at the beginning. Yes.
right, a lot of beginnings, but I think maybe I can just pick one thing. Maybe it's just very useful for guidance how to use in practice. So the question is from the Haijima. How do we know the true data generation prior? When you say the two priors should match, do you mean central mass overlapping? And I think it's just related to how to do the practice.
Yeah, so that's why I will say most of the time the analysis prior may be misspecified. So I think that's true. It's very hard to know what's the true process and the prior distribution for the theta. And of course, one way we can estimate it is, for example, in a specific disease domain or therapeutic, and you look at all the trial and look at their treatment effect. So that kind of provide an empirical estimate of the data generating or design prior. And from for the user prior and or analysis prior, and you can either match that. But when you match that, there's always a concern. Most times it's going to be informative. And how do you know it's unbiased informative prior, right? So I feel most of the time people may still use a kind of vague prior. And one way is rather than use a completed non-informative or very flat prior, you can based on, if you have that estimate of data generating or design prior, and you can discount, for example, you inflate the variance, make it a little bit vague. And in this case, at least you can increase robustness of your inference. That's what. And I think there is some question regarding how calculate PFDR and FDR. So that's a key question I want a quick answer. So that's essentially it's like defined in the guidance. So you do 1,000 simulation, and you look at how many times you have a positive study, positive trial, right? So essentially, it's the similar trial satisfy your Bayesian success criterion. For example, you have 20. And because for each simulation, you know what that true theta is, although it's random. But for each simulated trial, maybe sometimes it's -2, sometimes it's positive. It's 3. And based on your Bayesian criterion, you know the theta must compare with theta 0. If less than theta 0, that means it's not effective compared with control. It's greater than that, it's effective. So you can know what's the true status of positive or negative, and you just count among those a similar trial which satisfy the Bayesian success criterion, how many of them the theta is greater than theta 0, and you just do that calculation. That's a calculation of the PFDR. And for FDR, you just multiply that probability. So in your total 1,000 simulation, what's the probability the theta greater than theta 0, which is going to determine by the design or data generated by.
Yeah. I don't know if you still want to answer questions, Dr. Dr. Ken.
Yeah, I think it's okay. If the Zoom does not cut us off, I can stay longer. And
Okay.
for our participant, yeah, feel free if you need to log off. So I can look at it.
Yeah.
At least pick some. So it's a FDR, industry level metric. So I'm sorry, I'm not totally understand this question. So I'm gonna skip that. And if you yeah, want, you can email me about this question. I can answer through my email. And how to calculate the PFDR, and this I already described it. And here is a kind of clarification. I have a similar question. Is it a company level criterion for specific product or more of a cross industry wise metric? Okay, that's yeah, that's a very good question. For this FDR, again, because this FDR eventually going to relate it when we talk about the Bayesian OC, it's going to relate to the design parameter or data generating prior. So the question actually when it go to what does that data generating or design prior mean? Where that theta come from? It's just for example, I work on oncology. It's just theta, just the oncology theta treatment effect, or across all the TA. So in general, I will say that should be all the trial, all the possible study have been done in that TA. But if you want to generalize it, that's also, it's just a different interpretation, right? So you want to say, okay, so this theta, it's the treatment effect. Of course, it's random under the Bayesian thing. You have new compound, you can imagine that theta is kind of random, sometimes positive and negative. Sometimes you have a very good treatment effect, sometimes you don't have. And so what's the really that superpopulation you used to define your design prior? And I think that you can think in multiple level. If you want to interpret in a specific therapeutic area, so that's data going to include all the possible trial in that therapeutic area, and that's going to use the define the design prior. And once you have that, eventually, you're going to lead to the FDR. So it's and what's the interpretation of that value? And I think the other level of that question, when you say industry level, I think I feel overall FDR it's more kind of like regulatory side, maybe more interesting in that, right? Because given the approved drug, how many drug it's not effective. It's kind of like time and error. I think for time and error, it's mainly for regulatory focus because they want to control, in the long term, the false positive is like 5%. So it's Bayesian average type and error always smaller. This is oh yeah. So yes, that's true. Bayesian average type 1 error it's always smaller because the prior it's contains a value less than theta 0, but in frequentist we control it theta 0. But when you say smaller, it also depends on how you calibrate that cutoff C, right? So I think in here, what you really mean is when you calibrate the C, you calibrate under the condition theta equals theta 0. And then when you average across all possible theta, that's going to become smaller. But if different C, then that's different interpretation of results. So for FDR, do I understand it correctly? I think I can skip probably that question. It's very long. Okay, the next question. If the prior for the treatment effect is in the correct direction, should the type 1 error be automatically inflated? Also, should be try to keep the design and the next prior be the same? In the correct decision, should the type 1 error be automatically inflated? So for this question, it's of course, if possible, you should always keep make best to make the NS prior same as the design prior. But in practice, it's so hard because you never know most of the time what's the design prior looks like. And if the prior for treatment effect is in the correct decision, I mean, maybe you mean the positive decision. Should the type 1 error be automatically inflated? Yeah, okay. So that depends on how you define type 1 error, right? So in my presentation, when I define the Bayesian type 1 error or average type 1 error, I condition on the theta always equal or less than the theta 0. So in that case, you know, the question, the prior for treatment effect is in the correct direction. It's not applicable. But that's one of the reason I comment on Bayesian power. So Bayesian power exactly if you average across the whole range of design prior, then you mix the treatment effect in the positive side and negative side, everything together. So in that case, it's error or it's power? It's mixed of error and type 1 error and the power. So that's one reason I comment, you know, that the Bayesian power a lot of times is extremely hard to interpret because the average, the theta is positive or negative. That means it's very hard to mean what does that mean. It just means how likely at the end you can satisfy the pre-specified Bayesian success criterion. That's what it is. But it never related back to the truth. Can you provide some intuition for why one's analysis prior might differ from one's design prior? Does this imply additional information has somehow be acquired outside the trial, which causes you to update your prior belief after you have started a trial, but before you have collected trial data? Okay, so the design prior represent the truth, and in your TA, in your TA, and what's the true treatment effect looks like, how it distributed, and but for analysis prior, a lot of time we most based on what we know for that specific compound, or most time we just for convenience use non-informing prior to avoid potential bias. So that's why in many cases they are different. Okay, I think we can probably 3-4 minutes, 4 more minutes. Yeah. So next question is from Li Nin. Assume to design a pediatric trial use Bayesian borrowing approach and choose unblinded sample size re-estimation in case of underpower of this study because uncertainty during ongoing. Is it possible to still use conditional power to estimate the sample size, or it's better to use Bayesian predicted power to reevaluate? and how to consider type 1 error control in this unblind sample size re-estimation within Bayesian framework. Okay, so my response is you can use either way. You can use conditional power, or you can use Bayesian predictive power. But remember, the difference is Bayesian predictive power, it gonna account for the uncertainty regarding the theta, right? Because if during the interim, you look at the theta, you do power calculation, you conditional that theta. So in principle, Bayesian it's a more coherent approach because they're accounting for the uncertainty of that theta. And so once you use that approach, the really key question is how you calibrate it to satisfy certain error rate control or bias whatever control. So that's more of the question. And I think you can always calibrate your cutoff, right? Your success criterion such that your error rate controlled at a certain pre-specified value agreed with regulatory. That's the way I think you can use a Bayesian framework to do a sample size recalculation. And also different from a frequentist, which a lot of time rely on theory and asymptotic formula. And in Bayesian anyway, most time you have to do simulation. So in this case, you just calibrate in my example, you calibrate that cutoff C such that it satisfy the certain error. So you don't have to say always say average type 1 error or Bayesian type 1 error. I think that's going to be a discussion with regulatory authority, what exactly you want to control. Type 1 error or probability of a correct decision or probability of incorrect decision. So do you have a related paper for this presentation? Yes, I think it's in my slides. I'm going to share with you. How do we know the true data generation prior? When you say two prior should match, do you mean central mass overlap? Okay, so I already answered the first question. A lot of time we don't know the true data generation prior, but empirically we may estimate it based on a certain domain. And when I say match, I mean they are exactly same distribution, not only match the mean. Okay, sorry, what's the trigger of the interim analysis? Okay, so in my simulation and the evaluation, I assume the interim analysis is pre-specified, or you can, and also you can take a look at it any time because that's the intention I want. We investigate it. But the interim analysis is based on the same Bayesian success criterion as the design without interim analysis. So in other words, you can take a look at it any time. But again, because the multiplicity issue when the prior does not match, and you have to calibrate under your specific case setting. FDA did not mention in draft guidance one-sided or two-sided type of error should be reported in simulation. Sometimes two-sided error is not one-sided type of error. Would you please share your insight on this project, on this point? I don't think this Bayesian guidance focuses too much on type 1 error. It's especially I think the most significant development in this guidance it's more focused on Bayesian consideration, Bayesian OC. So I think this one that's okay. I think that's pretty much I answered the most. I see more and more questions at the end. Let me see. Okay, let's end 11:20. So okay, I can have a. Okay, I look at the last question posted by Haimin. So when we calibrate C to control PFDR, do we generate data according to pi g truncated at theta less or equal theta 0? The answer is no. So we directly generate data according to pi g. So remember, I think that's different from the frequent thinking about the power or type and error. In that setting, we control theta, we generate data. But in this case, we generate theta according to the pi g and then generate data. And in the simulation, we count how many similar trial it's positive study. And then we go back to look at the corresponding theta and see if that theta is greater or less than theta 0 to determine if it's a truly positive or negative study. That's how you use it to calculate the false discovery rate. And the other question, if we set the threshold of average type and error and the PFDR both at 0 to 5, which one will give less stringent decision rule? So that one, I think based on our simulation, seems maybe PFDR is more stringent. I cannot, actually, I don't know the exact answer. My intuition is it will depends on a lot setting, especially your data or your design prior, the pi g. So one thing we discover is it quite depends on the pi g distributed. When you look at the pi g, it covers all possible theta. So that means there is you can directly calculate a priori what's the true probability, the treatment in that population. Okay, in the treatment population, it's positive. So essentially, it's the probability theta greater than theta 0, but that integration calculation is regarding the pi g, the data generating prior. So that means in the truth or in what how many in drug actually it's tended to be it's the positive, and I think it will quite depend on that which one is more conservative. Can you consider the case that design prior is the same as the analysis prior, but consider different design prior in the simulation to get operating characteristic to accommodate the fact we don't know the true prior? Yeah, so that's the one point that I highlighted, in the conclusion slides. It's important that we do sensitivity analysis. That's also described in FDA guidance, because the truth is we never know how different they are. So it's better you can do a different sensitivity analysis. How to calibrate type 1 error? But again, I don't know if this means Bayesian type 1 error or frequent type 1 error, but the basic principle is the same, right? So in the Bayesian type 1 error, you just when you simulate data, you every time you draw the theta from the support where theta less than theta 0, and you do simulation and count in those study and those negative study how many times you claim success. That's the type 1 error. Last question I see is how to fix the potential increase in type 1 error for the multiple look Bayesian design, frequentist adjust the critical value. Okay, that's a good question. So there's two ways, at least the two most straightforward ways to control multiplicity for Bayesian design. If we, in the case, the two prior don't match. One is you can adjust the cutoff C in the same way as a kind of frequent thinking to compensate the inflation. So the other way is you can use a more pessimistic prior. That's the user analysis prior to compensate that inflation. But in general, I will more prefer calibrate the cutoff, because cutoff does C anyway. It's a value we should calibrate to get a good OC. But for the prior, a lot of time it have a specific interpretation, right? And it's very hard to say, oh, I tweaked my prior. It's just to compensate a potential impact due to the prior misspecification. Yeah, and for the multiple look choose of C, C must be just any guidance on choose C. So that one is you choose a certain metric. For example, if you want to control FDR, and let's say you want to control FDR at 5%, and you can through the simulation and do a grid search, find a C such that FDR is controlled when you have multiple look. Okay, I think I pretty much.
Do you want to stop here?
Yeah, I think we can.
I think.
We can stop here. But yeah. So if anyone have a follow-up question, you are very welcome to send me email. I'm happy to answer those questions.
Okay, thank you very much, Professor Yuan. Excellent talk and also very exciting interaction with the audience's questions. It's very, very helpful. I think we just want to you have more method development and some other paper clarification, operational and theoretical to the broader audiences. Yeah, and also thanks for our people joining today. And just see you next time on the
Yeah, also I
ISBS.
want to thank you to everyone attending this session and also stimulating a question discussion. So that's always very helpful to me. Maybe there's something wrong or not well written in my paper. So any comments, it's welcome.
Okay. Thank you. Thank you, Professor Yuan, everyone. Bye-bye.
Bye bye.